This part we are going to learn the classification of bacterial. Bacterial can be classified based on two main characteristics. First, we are going to look at the bacterial cell shape. The first one over here is round, and we call it as coccus or cocci, which is the plural form. If singular, then we say coccus. It's round-shaped cell, spherical shaped. While if the cell shape is like a rod, then we call it bacillus, rod-shaped, as shown in this picture. If it's like a spiral, the spiral shape like is twisted, we call it as spirilla if it's plural, and spirillum if it's singular. Another one you can see here, the cell shape is a bit like a comma. So this one is what we call vibrio. So from the name of the bacterial, if it say vibrio or spirillum, bacillus, coccus, all this tells the shape of the bacterial. And when you look at its arrangement, if it's only two circles together, then we call it as diplococci. We know it is round, and we know it has two round-shaped cells together. If it's a chain of the round-shaped cell, we call it streptol. Cocci tells the shape of the cell, it is round, and streptol tells that these cells, they are in a chain. If it's in a group like this, then we call it as staphylococci. Another way to classify the bacterial is based on gram staining. So gram staining, it is a technique used to differentiate bacteria based on the structure of the cell wall. Before we look at the structure of the cell wall of the bacterial, let's understand how does gram staining work. First, we need to put a purple dye. After putting a purple dye, we will put iodine. The combination of the purple dye and the iodine, it will form a large compound that can be trapped in the bacterial cell wall. And then we put decolorizer, which is alcohol. Alcohol will wash away this purple dye molecule if the cell wall is thin. But if the cell wall is thick enough to keep the purple dye, then adding alcohol, it will not wash away the purple dye. And the last one, we have a counter stain, which is a dye of another color, is a red dye called safranin. What is the purpose of adding safranin? If the cell wall of the bacterial is thick, then it will retain, it will keep the purple dye, and even we add alcohol, it will not wash away the purple dye, so this thick cell wall bacterial will remain purple, the first dye we use. But if the cell wall of the bacterial is thin, then when we use alcohol, it will wash away the purple dye. When this purple dye is washed away, this bacterial cell will take up the counter stain, which is red, and that's why the bacteria with a thin cell wall, it will appear red. And from this color here, based on gram staining, we can differentiate what type of bacterial they are. And this is all because of the cell wall structure of the bacterial. We can classify bacteria based on gram negative or gram positive. If the cell wall is thick, then it can retain, it can keep the purple dye, which is the first crystal violet dye, and that's why gram-positive bacteria will be stained violet or purple. Even after we wash the bacteria with alcohol, it does not decolorize the bacteria because it has a thick cell wall to retain the purple color of the dye. thick layer of cell wall made of peptidoglycan. And if you compare it with gram-negative bacterial, you can see that the peptidoglycan cell wall of gram-negative bacterial is very thin. And because it's a very thin layer of cell wall made of peptidoglycan, so it can't retain the purple dye. That's why if we wash it with alcohol, it will wash away the purple color, and it will only get stained the last color given, which is the color of the safranin, a red dye, or sometimes it looks pink. From the color itself, we can tell which bacterial is a gram-positive bacterial, that's why purple, which is the gram-negative bacterial, that's why it's red or pink. And all this is because of the cell wall structure of the bacterial, whether it has a thick peptidoglycan layer cell wall or whether it's a thin peptidoglycan layer cell wall that cannot trap the purple dye. What else is different between the cell wall of gram-negative and gram-positive? You can see here, gram-negative bacterial has two layers of phospholipid bilayer. It has an extra outer membrane. And because of having this outer membrane, it makes the gram-negative bacterial more resistant to antibiotics, which means that the gram-negative bacterial will be harder to be killed by the antibiotics. Other than that, on this outer membrane, it has a kind of structure that we call the lipopolysaccharides. And lipopolysaccharides is combination of lipid and polysaccharide. It is toxic. It will make the gram-negative bacterial more pathogenic or more virulent.